All right, here's part 3 of chapter 1, looking at the major differences between prokaryotic and eukaryotic cells as a means of how cells are characterized and organized. First off, the smallest unit of life is considered the cell, and then you work your way up from cells to tissues to organs to beings, and regardless of where you're at on that spectrum, the cell is the smallest unit of life in its entirety. Alright, so we've mentioned the two basic categories are prokaryotic and eukaryotic. And these are, it's just written out of what I mentioned in part 1, I believe. Prokaryotic cells or organisms are unicellular. That means there is just one cell that makes up a complete organism. Alright, lacks a nucleus, and the two main categories are bacteria and archaea. Alright, remember the archaea are really what's considered the extremophils. But however, there is data to suggest that even in our gut flora, in our intestinal tract, there are a few species of archaea, just in us. Alright, then eukaryotic organisms, also previously mentioned, can be unicellular or multicellular. There is a distinct nucleus, and there's 4 main types of eukaryotic organisms: the animal, which is also us, plants, fungal cells, and protists. Alright, a person named Carl Linnaeus is considered the father of taxonomy, and this is a way of using names to classify and organize organisms. And he developed what's called the binomial nomenclature system. It's a 2-name system where the genus is first and it's capitalized. The species is the second name, that is always lowercase, and the 2 names together create the scientific name, and these are always italicized. And if you can't italicize them, such as you're just writing them in your notebook or something, you can also underline the word, and that is a proper way to get around not being able to italicize. So for example, you have Escherichia coli, right? Uppercase for the genus name, Escherichia, and lowercase for the species name, coli. And a lot of times we just simply refer to that as E. coli. That is what it is referred to in just the general news. Not a lot of people know that Escherichia is what the E stands for, but that is the proper scientific name, just like Homo sapiens would be the proper name for humans, Homo being the genus and sapiens being our species. Okay, then you have different ways of this taxonomy and how to use that. And the major domain is the broadest group of organisms. And there are 3 domains: bacteria, the archaea, and eukarya. Right? Another thing to mention with the archaea is there aren't any known pathogens. Because they have such extreme environments, there aren't a lot of other living material in those environments, so there isn't anything for those organisms to actually prey upon to cause disease. Alright, so table 1-2 looks at the domains. Alright, you have the archaea, bacteria, eukarya, 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 eukarya. But then you have the kingdom. And this is what's considered the 6-kingdom classification system. So you have the archaea and the bacteria as 2 kingdoms, both, in this case, both are prokaryote, but you have the 2 different groups, domains, kingdoms. In this case, they overlap or are redundant. And then the kingdom of fungi, plantae, animalia, and protists. Right, and then all 4 of these are eukaryotes. Now there's also the strain. So you have the genus and species. as seen here, E. coli. But there's also strains used to recognize genetic variants of the same species. And in the news, when you hear about E. coli and E. coli breakouts, that's usually referring to E. coli 0157. And that 0157 is a specific strain that has a particular genetic makeup of the O antigen. A lot of information there, but there's a lot of strains out there. Sometimes the strains are pathogenic, sometimes they are not. How do you get new strains? You have mutations that occur at the DNA level, and there's also gene transfers, where genetic material from one cell can get transferred over to another cell. All right, host microbe interactions. All right, we are interacting with many microbes all the time, daily, constantly. And there's different ways that we can refer to those host microbe interactions. The symbiotic relationship exists when there's two or more organisms that are closely connected. Right? We have a symbiotic relationship with bacteria. The bacteria that live on us and in us, obviously it's a very close relationship. Most of them help us, we help them, we give them food, we give them a home, and in return, they either do nothing for us or they actually help us out. But there's a couple of different symbiotic relationships. And most of them are going to be in these bottom two options. The commensalism, no perceived benefit or cost to the host. So if you look at it from the standpoint of the bacteria, many of the bacteria that live on us are commensals. And that's sometimes a way to refer to the bacteria on us. They're commensals. We don't actually have any benefit, and it doesn't cost us anything to have those bacteria on us. Then there's mutualism. The bacteria are going to help the host. So for instance, E. coli, if it is the right strain, if it stays in our GI tract, it will actually, in the process of metabolizing the food that we give it by eating and having food in the intestinal tract, some of its metabolic byproducts are actually helpful for us, like, for instance, vitamin K. All right. The vitamin K that we use is actually made by bacteria in the gut, and so then our intestinal lining can absorb the vitamin K and our bodies can use it. And then, of course, there's parasitism, all right, where an organism will be a parasite. It hurts the host. And thankfully, we have much more of this commensalism or mutualism than we have the parasitism. But one example of the parasitism would be the E. coli, that when it's in the GI tract, it's going to be mutualism, but if it makes its way to the urinary tract system, then it's going to become basically a parasite. It's going to cause disease. Alright, so pathogens have a parasitic relationship. They are a pathogen. They cause disease. Now, in all reality, the term parasite is generally used to describe helminths. Those are pathogenic microscopic worms, sometimes macroscopic, and then protozoans. All right, if you just think, when someone says they have a parasite, right, I think of a eukaryotic organism, the helminths, the protozoans. Whereas if it's a parasitic relationship, then it's just a pathogen causing disease. Different ways that bacteria interact and actually protect themselves from the environment is to create a biofilm. Most bacteria are what are considered to be planktonic bacteria. Those are free-floating, they're really doing their own thing, and life is okay. But if the bacteria creates a biofilm, The biofilm is a community of bacteria, and generally it's quite sticky, and it's made up of a single, but more than likely a diverse microbial species, a lot of different bacteria. And there are cells that seed a biofilm, and they make adhesion factors, which helps the process start of being able to create this layer of bacteria. Right? Those cells that are going to be there initially secrete a material that helps to protect the bacteria and allow them to grow. And then you have multiple layers of this occurring all the time. And in the end, the bacteria within all of these different layers are protected. And so then it's hard to get rid of them. All right, and sometimes microbes in the biofilm are going to be released as free-growing planktonic cells. And so here's an example of what's going on. There's attachment, and we'll look at this in more detail further into the semester. Here's the surface. So if you think about your tooth, all right, our mouths are a very dirty place. Bacteria can attach to the surface of the tooth, and once they sit there and stick, then they're going to start producing this matrix, this brown stuff. All right, and that will continue to grow, and occasionally bacteria can get out of that biofilm and move somewhere else. And this is a scanning electron microscope picture showing dental floss with biofilm scraped from the teeth. So the teeth are the perfect example of a biofilm in the human body. You've got all that slime that you can pull off your teeth, and sometimes it's pretty obvious that it's a chunk of food, but when it really is just that slime that you can scrape, and you have issues with the plaque, and then that hardens, and you get the tartar buildup, that's a biofilm. And you've got all of this a mix of this matrix stuff and the bacteria that's found in the dental floss here. All right, so real fast, we have normal flora, sometimes referred to as normal microbiota, and these guys have an important job. All right, they train our immune system. Right, we are a complex organism as humans with a mix of bacteria in many places, but then also our immune system that we want to train so it works properly and it doesn't overreact. And in the cases of it overreacting, then we can develop different diseases, autoimmune disorders, inflammatory bowel disease, those type of things. So the bacteria get to train our immune system and say, hey, this is normal, don't attack me. They produce vitamins for us, like I mentioned, and they also help digest our food. Right? The bacteria are really good at breaking food down, and more data is coming out about how bacteria can actually impact our mood as well as our brain function. Alright, so you can look at all the different bacteria that are present as our normal flora, and this information came out from the Human Microbiome Project that looks at some of the different areas of the body where we know that bacteria are present. So, just for example, in the intestinal tract, 40,000 species of bacteria live in our GI tract. Upper or mouth, the pharynx, upper respiratory tract, 600 species. So our mouth is a very dirty place. Right? They've got the lungs here colored in the green, but the lungs are actually the lower respiratory system, so you really shouldn't have bacteria in your lungs. All right, how do you establish normal flora? This is going to occur as soon as the infant is brought into this world. All right, there's data that shows that babies who are born vaginally versus C-section have a very different normal flora, and then they continue to develop their own normal flora throughout the first few days of their life. transient bacteria, they're there for a little bit, gone.